I'm doing the first one.
All right, it's recording. All right, I got odds, you got evens?
Yep.
Awesome. Welcome to today's Bio 285 Cell Cycle 1 review presented by yours truly and Lillian. Yeah. Topic 7.
All right, so this is our cell cycle 1 topic, which, I feel like you guys are probably pretty familiar with. So this one might be a little quicker, and then we can do problem set questions or exam questions after. So just as like an overview of what we are looking at for the cell cycle, the only phase that we might talk about that maybe you haven't talked about in your other classes before that you might not be familiar with is G0. So that is kind of removed from the cell cycle, or maybe a subset of G1. And G0 is when you are in a non-dividing state. And that could be because you just went through a full cycle and you're waiting to restart, or maybe the cell has just removed itself from dividing. Some fully differentiated cells will always be in G0 and they'll never leave, but some can come out of G0 and go back into the cell cycle. And when you leave G0, you go into G1. So that stands for gap, but a lot of people remember it by G for growth, because that is when our cell is doing its prep for DNA replication. So it wants to make sure that it's adequate size, it has the amount of nutrients that it needs, no mutations, whatever. And this is also where we have our restriction point, or start in yeast, but we usually call it R, the restriction point. So after we've done all of that, if we don't get held up at the restriction point, passing that is when we're fully committed to the cell cycle. So after that point, the cell is either going to divide or it'll undergo apoptosis if it doesn't have the stuff that it needs. So it's really important that during G1, it does make sure that it has what it needs to go through with division. After that, we go into S phase, synthesis, and that is when we replicate our DNA. And we talk a little bit about this. A lot of textbooks won't even bring it up, but there is like an end of phase S phase checkpoint where we just make sure, again, that everything is going smoothly. The cell cycle is really heavily regulated at almost every step because we want to make sure that we don't have DNA mutations when we're dividing into a new cell, because that's when you get diseases like cancer. Then we have G2, which is pretty much the same as G1. We're preparing to segregate our newly synthesized chromosomes, and we want to make sure that our cell is roughly doubled in size, because when we go through cytokinesis, you want to make sure that you have like 2 fully formed daughter cells. And then we have M phase, which is mitosis, and that, I guess, that's separate from interphase. So all of our other phases up to this point have been a part of interphase, and then M phase or mitosis is kind of its own thing. So we go through prophase, metaphase, anaphase, telophase, and then cytokinesis when we separate into our 2 daughter cells. So that is an overview for you.
All right, so first step, G1, I think it's kind of underrated because people usually talk about like most of the other, like, oh my God, M phase is so important. There's all these different like steps I want to talk about, and like it gets kind of like forgotten. It's like pretty important, I would say, because like Lily was talking about beforehand, it's either you arrest it or you don't, and if you don't arrest it, you're going through with it, and it's either you're making it through as 2 new daughter cells or apoptosis. So I mean, it's pretty big deal. And so the cells are still in G1 after restriction point. It's important to note that cell growth processes are, they take a little longer. They're not like the shortest steps. It's not just like, oh, here, take some whey protein and then grow. No, the cell growth process takes a little bit. And so you need a growth signal to start cell division, and so we learned about this in the RTK RAS MAPK pathway topic 5, topic 5. So like Laura likes talking about old concepts always come back. What's it called? Microtubules is also pretty important once we get a little bit deeper into the later stages of the cell cycle. And so it's just don't forget about the older topics. They do come back. And so in this step, cells are checking for DNA damage, they're checking for correct size, checking for energy, checking for nutrients. No 2 daughter cells are always going to be 100% like the exact same. They're not going to, you don't have like that many mechanisms to be able to like say, oh, this mitochondrion has to be the same size as this other one or else it won't. Like the process, yes, is complex. Yes, it has all its different steps to it, but you're not gonna get like two daughter cells that are 100% like every single thing similar to each other. And so you got to check for different nutrients. You got to check to make sure all your enzymes are working, all your different processes are working for perfect cellular division. And if the preconditions are met, then these cells will pass through the R point, and then it's time to get going. And it is a very, very critical step, because if one little thing is off, we will learn moving on about different problems that can occur if premature R checkpoint is like done through. And so Laura also really likes asking questions about if this happens, then would the cell pass the R checkpoint, or if the cell passes the R checkpoint prematurely, what would happen? And so it's good to keep in mind that there's different processes that we need to keep in mind and make sure that we understand, so that if she does ask questions, we're able to answer them. And so after the cells go through the checkpoint, there's two different options that we have mentioned beforehand. It's either you divide two new daughter cells or apoptosis.
If I can just add on to something that you said, you were talking about like what would happen if the cell went through the restriction point prematurely or something like that. For this exam, I think that kind of question is especially relevant, especially because we have the pathways in topic 5. So be comfortable with looking at anything that we've talked about during this section and saying, if you break this one aspect of it, what effect will that have on like the bigger picture in the cell? That is definitely a question or a type of question that you're probably going to see on the exam. So a good way to do that is like if you're looking at a diagram of something, if it's like your RAS RTK map K, if you cross out your SOS, or in the cell cycle, like if you think about what would happen if we skip this, what are we missing, then that is something that she might ask you on the exam. Okay, so S phase is pretty straightforward. We're replicating our DNA. We go into the details of this at the very end of the semester, so you don't really have to know much about this other than we can go through our little checkpoint. We can temporarily halt our S phase if we sense an error in DNA replication. And then what will happen is if we can fix the error, then we will resume S phase. We'll keep going through until we hit G2. Or if we can't fix it, then the cell will go through apoptosis, because we do not want to let the cell sit for too long with mutations accumulating. We don't want to risk having daughter cells with potentially harmful mutations. So.
Any questions so far? No? All right. Cool. So now picking up steam, we're moving on to G2. The G2 is pretty important. Here, like Lily was talking about, we're going to learn a little bit more about different types of DNA repair and DNA check mechanisms. Right now, what you basically need to know is that the G2 stage is basically preparation. So you're prepping for M phase. And so you're making sure that your cells are the appropriate size. You're making sure that the different processes that need to happen before metaphase is happening, or sorry, not metaphase, prophase are happening. And so it's also pretty common. You'll if you like read any article online, or if you like look into like different YouTube videos, people are going to talk about the G2M checkpoint. And so it's a pretty important checkpoint. It prevents cells from entering mitosis. And so they check for DNA, if there's any mutations, if there's any damage, anything that needs to be spliced out. And if the damage or mutations or whatever is too harmful, then it arrests the M phase. And so if all the checks aren't checked, I guess you could say it like that, then it doesn't proceed, and the cell also goes to apoptosis.
Okay, so now we have M phase, and this is kind of where our active process of division is happening. So I'm sure you guys are all kind of familiar. You should know basically what happens in each of our phases: prophase, metaphase, anaphase, whatever. But what we really want to focus, especially for the later units, is our spindle spindle assembly and chromosome segregation. So our spindles are the microtubules that we were talking about during topic 6. Basically, our microtubules are going to form out from our organizing complex, and they're going to reach out and grab our kinetochore of our sister chromatids. And something really important that happens during M phase is that all of our sister chromatids have to have a spindle or a microtubule attached at their kinetochore, and they make really specific binding interactions. And you have your two sister chromatids being pulled essentially from opposite sides by the two ends, or your two poles, your spindles coming out of them, and that puts tension on either end. So what they think is that when you have all of your chromosomes lined up during metaphase, you have all of your spindles attached, the cell senses that you have equal tension pulling your sister chromatids towards either pole of the cell, and that's how it knows when all of its correct attachments are made. But at our spindle assembly checkpoint, which is our really important M phase checkpoint, we really want to make sure that all of our spindles are attached to our kinetochore, so when we separate, we don't end up with like a non-disjunction or, you know, chromosomes getting lost. We want to make sure that we're dividing with a full set of DNA and nothing extra. And during any of our cell cycle checkpoints, if we halt for too long, if the cell spends too long trying to fix a problem, then either way, it's going to go through apoptosis. So it basically has a limited window before some other mechanism in the cell decides like, okay, whatever damage here is not being fixed fast enough. So this is all pretty tightly regulated. We want to make sure that we're ending up with complete daughter cells. Go ahead.
So does apoptosis happen like if any checkpoint fails?
If any checkpoint fails, then yeah. Except maybe G1, if you don't have like enough nutrients, then it'll just go back into G0. Because at that point you haven't initiated anything. So if it just needs time to, you know, keep growing or to bring more nutrients in, but if it's, especially if it's a problem with like mutated DNA, then at that point, if we stay for too long and the problem isn't being fixed, then we'll go through apoptosis.
Yeah, also, I don't know, I'm kind of in a sense of trying to figure out like the steps, because you might draw your pathways and you might draw your cell cycle and have it on the top, like on the top of your mind the day of the exam. I also like having to myself the ability of like naming the M phase steps. And I think the way I memorized it, they kind of like lead into each other all the way through anaphase. And so there's different sources online that if you look into it, we'll have a step between prophase and metaphase, which is called prometaphase. And so how I liked looking at it, like when I was studying for this, when I was a little less familiar with the content, I used to just go pro, meta, meta, ana. And so that was my way of like memorizing like the first 3 steps. Telophase is a little bit different, but it's also I would say a little bit easier to memorize because the cell is like huge at that point, because it's like, you know, about to divide and like everything is like duplicated and like the cell is like bigger. And then cytokinesis is the last step. It is the only step that doesn't end in phase, and it for me it's the easiest step to like know because, you know, it's 2 different daughter cells and it's, the process is over. But I would suggest that you guys have some sort of initiative to try to memorize the steps. Even if you don't memorize like what every step 100% does, have it in your mind that like, oh, it goes pro, meta, meta, ana, and then telophase, cytokinesis. It helps, especially if you're like looking at a question, you're like rushing through the exam, you're like, you know, under pressure, having to finish, all the different thoughts in your head, and maybe you're blanking out because it's just how the human brain goes during exams. If you have that little bit of knowledge that you can like rely upon, and then you're like, oh, so this happened, and then, you know, it becomes a little bit easier.
If you're having a hard time with the order, I always said PMAT in my head because, you know, those are the first initials. I don't know if that helps anybody because it doesn't really mean anything, but that's what I did. Okay.
All right. Gee, that's an odd slide.
I remember. So now we're going to go through a little more of the cell cycle control systems. So One of the main mechanisms that we talk about is phosphorylation and dephosphorylation. So kinases, as I'm sure you're familiar with at this point, are proteins or enzymes that phosphorylate another substrate. Usually it's going to be a protein, almost always, sometimes it'll be a lipid. And phosphatases are the opposite of a kinase. They remove phosphate groups. So something that we usually think about, especially in terms of like GTP versus GDP, is the phosphorylated version being active. But in this unit, we are going to see an exception to that, which is V1. So something to be aware of is that there is such a thing as a deactivating phosphorylation. So usually phosphorylation is your on switch, but that is not always the rule. There are going to be exceptions to that.
That also happens to BAD in the cell survival signaling pathway.
Yeah.
So if it helps, there's another example that we've already gone over.
Yeah.
Um,
So whether it's an on switch or an off switch for that particular protein, phosphorylation is just going to be one of our primary control mechanisms. And the kinase that is going to be doing a lot of that is CDK.
all right, CDK, cyclin dependent kinase. We're gonna hear a lot about this bad boy over the course of the next few weeks. So they are, as Laura said in class, they are the master cell regulators. They are very important for different cellular functions, and they are everywhere. And so they are serine and threonine kinases. So they add the third phosphate group to serine and threonine R groups. And who's feeling brave? Who's feeling brave?
No, wait, I'm not.
Okay, so what do we need in order to phosphorylate? Yeah, but in the R group, sorry.
No, you're good.
Anybody else? Yeah. Well, in this case, you're activating serine or phosphorylating serine and threonine. But on the previous cases that we've been studying on the tyrosine kinases, there's something that's common to all three of those amino acids, and that is the presence of that hydroxyl group. So you need that OH in order to phosphorylate. And that's just something that keeps coming up. It might be a final question. It was a final question in the past. Let's make sure we're remembering that. It's been a long time, and so let's not forget that. You need hydroxyl group in order to phosphorylate.
So like,
It is not an ionic bond formation, as compelling as that might sound for you to write on the exam. It is not
yeah.
an
So you
ionic.
would have had an H here, but instead you're going to attack it with your phosphate group, and that is where you're attaching that hydroxyl right there.
So do not feel compelled to write it's an ionic bond formation. That is, yeah. Huh?
That's a crazy.
Yeah, couldn't beat me. Anyways, so CDK regulates different things that we were talking about. It can regulate cyclin levels. It can regulate two different types of phosphorylation. And like Lily was talking about, this step is very important. It activates CAC. But then it also acts in a way of phosphorylating V1, and then that actually deactivates it. Sorry. Yeah, no. I worded that wrong. But the process of phosphorylating and dephosphorylating in this case, or phosphorylating on both cases, first case activates it with CAC, and then V1 deactivates it. I don't know. Do you want me to reword that? Can you reword that? That was awful. And let me go back. So 5 different things that control CDK. First one is the cyclin levels. You are, like the name says, you are dependent on cyclin levels in order for the kinase to, function. So the phosphorylation of CAC, the phosphorylation of CDK through CAC activates it. And then the process of phosphorylating V1 comes in and deactivates CDK. Something else that works in the regulation of CDK are the CDK inhibitors. And the kinase can also be dephosphorylated by different phosphatases. And so for active CDK to happen, or to exist, you need two events. And first is you're gonna bind cyclin. And then the second step is you're gonna be phosphorylated by CAC, which is CDK activating kinase. And so it's a two-step thing. Laura might ask, oh, in the case that cyclin has low affinity for CDK, like what happens and why? And then you should be able to explain, oh, since cyclin CDK complex is important, and in this case, cyclin is unable to bind to CDK. And then in this case, it's unable to bind, then it will not be able to be activated and not be able to perform its functions. And then you can talk about whatever function you want to talk about.
For like the phosphorylation, like that we said, will deactivate is like the V1. Since it's actually the V1 or
V1 kinase, yeah, but we just call it V1.
V1, okay. Is that the only example of that?
Of
Like
Phosphorylating-
deactivating?
so in the cell survival signaling pathway, BAD, which is the protein that sequesters BCL2, is phosphorylated, and that deactivates it, or deactivates its function of sequestering BCL2, which then leads to cell death not happening. But that's just another example. I understand the confusion. It sounds counterintuitive. Like everything we've been talking about has been like, oh, phosphorylation activates something. For these specific cases, just have it in like your.
During exams, you have to like know which one of these are.
Since they are two important examples and two like crucial things on either like general field of like topics that we are studying, I would recommend writing it down and having it, because like it's an essential step on the cell survival signaling pathway, and it's an essential thing for cyclin dependent kinase. The fact that it gets deactivated by another kinase that was phosphorylated is like a very important aspect of it.
I would also say though, if any of you are visual learners, because this very much applied to me, sometimes it's hard to conceptualize this just looking at it written on the slide. I would really recommend before the exam going back through her slides, because she has some really good diagrams, and sometimes I think it makes more sense to see, okay, we're adding a phosphate to this and now it's whatever function it was doing is not happening anymore. I think visualizing it is really helpful for these. It was
Also-
for me.
oh,
And
yeah.
drawing the pathways.
Go ahead.
Kind of bond.
It's a covalent. Yeah.
Yeah, I'd also recommend reading through her topic notes. Her topic notes are like very well explained in like a word setting if you're not a visual learner.
So, yeah, that's true. Also look at her topic notes, because she writes those herself, so they are what she finds to be the most important. But your post-translational modifications, they are reversible covalent attachments.
Anything else? Questions, comments, concerns? Going too fast, going too slow? No? All right.
Okay, so now we have cyclin. Cyclin is not a kinase. A lot of people get that mixed up. If this trips you up, like CDK literally stands for cyclin dependent kinase. So just remember that there's only one in the pair. Cyclin is not a kinase, it binds to the kinase. So we need cyclin for CDK to have its kinase activity. Yeah, sorry, I'm gonna move closer to the mic. Yeah, so cyclin activates CDK as a kinase, and also it gives it substrate specificity. So what that means is when they are bound, that's kind of what gives instruction to CDK on what it should phosphorylate. So I don't know actually the specific mechanism of that, but maybe it contributes to like an active site when they're bound together. It adds to like a binding interface, but we need cyclin to tell CDK what to bind to. So unlike CDK, our methods for regulating cyclin are pretty limited. We have transcription and translation, so we can make more of it when we need more of it, and we have ubiquitin-mediated proteolysis, so when we don't need it, we can degrade it. That's about it in terms of regulation. And we talked about this on whatever day we had class. Was it yesterday that we watched this video? we watched a video about like the T loop, and you don't need to know the mechanism for this on the exam, but oh yeah, that's right, that's what it is. So our cyclin pulls back our T loop on CDK, and that is what exposes the binding site. So you don't have to know the specifics of that. I don't think she asks about that, but if it helps you in your brain kind of remember how that goes, that is what the deal is there. And I do also think that that movie is really helpful if you want to rewatch it, if this mechanism doesn't make sense to you, but you don't have to know it in great detail.
All right, so continuing, cyclin, CDK complex, CDK cyclin complex, whatever you want to call it. Very important, very important. CDK cycling is a very broad term. There are different types of cyclin that are translated in different stages of the cell cycle. And so do they need to know each specific
No.
type? Okay, so according to Lily, don't quote me on it, you don't need to know. Oh, okay, you can quote her. You don't need to know every single specific type of cyclin that is present. But so why is this important? Because CDK functions during the cell cycle with different cyclin partners. And so each specific cyclin has a specific function in order to aid CDK to know where it needs to go, what it needs to do. And so again, reiterating it, how do you get the same CDK functioning throughout the cell cycle and targeting different proteins? Cyclins act like a guide, and so that's how CDK knows what it needs to do. And so the example that's on the slide, cyclin E, which is at the bottom right of the cell cycle over there. If you couldn't find it, too small writing. It helps CDK target specific proteins after the restriction point. And so maybe Laura will ask, oh, like if levels of CDK expression are low, explain how like different functions could be performed by the same poor CDKs that are overworked or something like that. And so that's how you're able to say, oh, because of the different existing types of cycling, you're able to get one single CDK and have it perform multiple different functions. And so cyclin levels rise and fall during the cell cycle, depending on what type of functions need to be performed, which is why you're seeing that weird squiggly looking graph at the bottom right of your screen. And so the levels are solely controlled based on their concentration. And so as you can see, G1 cyclin is the one that stays active the longest. Whereas the other ones are kind of like more specific. S cyclin is also pretty big, but that's going to differ on the different functions and the different things that happen throughout each of the different steps of the cycle. I should probably talk about ubiquitination. Sorry. Also, cyclin levels can you can overproduce cyclin, but that also becomes an issue if there's too much. And a process of getting rid of the excess cyclin is ubiquitination. Have we gone over ubiquitination class?
We've talked about it briefly. Ubiquitin is kind of like a phosphate group. It's another functional group that can be covalently attached as a modification, and that just flags for it to be sent to the proteasome. Do you have a question about
More about this,
yeah, you don't really need to know anything about it other than it's a post-translational modification, and it's just like a flag for another carrier protein to pick up the marked modified proteins at the proteasome.
And then it gets degraded.
So it's a degradation signal. Go ahead.
So what's the degradation after?
All throughout the cell cycle.
Whenever we don't need that protein, we'll tag it with the ubiquitin, and then it will be degraded.
It's not only for cycle. It happens for
Right, yeah.
It's for like everything.
So the same thing will happen, I think, is a UPR. It happens in like the ER too, or maybe heat shock response, but it happens to different proteins. Just generally in the cell, if something is tagged with the ubiquitin, it means that it's going to be sent to the proteasome.
Questions? All right.
Okay. Yeah. So cyclins, as John just kind of talked about, we see in this diagram in our cyclin expression cycle, we have our G1 cyclin, our G1S, whatever, that change in their concentrations throughout the cell cycle. So they are specific to phase. There is a G1 cyclin that is active during G1. There's an S phase cyclin that's active in S phase. But CDK is not. We have basically just our CDK that functions consistently throughout the cell cycle. And then depending on the depending on the phase that it's in, it has a different cyclin as its binding partner. CDK, we have activation by CAC. So that is an activating phosphorylation. We need a couple of things to happen for CDK, I guess, to be active. We need to be phosphorylated by CAC, and then we have inactivation by WEE1. So that was the example that we were talking about earlier. And as I mentioned before, phosphatases, if we are taking off a phosphate group, it's just the opposite of a kinase. And we can also regulate with CDK inhibitors. So those are, we were talking about G1 regulation mechanisms. We see those a lot in response to DNA damage. Those are different than like a ubiquitin tag because they're not going to send it to be degraded. It's just going to kind of hold it in an active state. And CDK is a lot of times the target for inhibition, or like the molecular breaks. Laura calls them breaks a lot. So it's okay if you guys, it's a small group, but does anybody have ideas on why this might be a really good target for inhibition when we're regulating the cell cycle? So what is the nickname, I guess, that Laura has given to CDK? Go ahead. Right, yeah, it's the CEO of the cell cycle, it's the master cell cycle regulator. So basically, if we're looking at the cell cycle as like this cascade of events, our CDK is at the very top, and CDK activity allows all of the things underneath it to happen that enable the cell cycle. So if we put an inhibitor at the very top of this big cascade and we stop CDK from doing whatever it's going to do, that is a really fast way to put the brakes on the entire cell cycle instead of targeting a specific substrate that's down here. Because down here, there might be 20 different things that you have to target to totally stop, whereas you could just go for CDK and kind of put the brakes on everything, especially if you really needed to, like if you had DNA mutations, whatever.
Speaking of DNA mutations.
Yeah.
That's a great view. Okay. So very, very important. I'm also fascinated by this topic. I think it's very interesting to study. So cell finds a DNA mutation in G1. What happens? Well, P53 gets activated. All right. So P53, transcription factor, tumor suppressor, and it typically responds to DNA damage. There is a question on the problem set about this, so if you do have any questions about it, ask. And so what does P53 do? So senses the DNA damage, and so it activates the transcription of B21. I forget what B21 is, but there's like a name to it. Yeah, there's a name to it, but yeah, there's a name.
Oh, you're right, there is.
Yeah, there's a name.
Oh,
but so P21 inhibits CDK, and without CDK being active, your cell cycle activity ceases because, like Laura likes to say, CDK is very important and is the master cell cycle regulator. And so if DNA damage is irreversible, P53 can also signal for apoptosis. But I would like to put an emphasis on this. P53 is very, very, very well regulated. Cells always keep an eye out to see P53 function. Not really keep an eye out because they can't see, but P53 is very, very, very common in cancerous phenotypes. Maybe there are viruses that trigger an activation of it, or there are different types of infections that can lead to P53 not being able to perform its job. P53 mutations are widely studied in oncology, and that is because once P53 becomes unable to perform its function, cell proliferation happens at crazy amounts with, it can be DNA damage leading to, malfunction. It could be DNA damage leading to, apoptosis. It could be DNA damage leading to, overproliferation of, cells that do not die. And that is what we come to know as tumors. And so, I would like to put, again, emphasize P53 is a very important, protein in this, and just make sure that we're understanding how P53 works, how P21 works, why ceasing CDK activity leads to, a halt, and why P53 is so important. Anything you wanted to add?
Yeah, I am actually gonna add on to that. I don't remember if it was like grading problem sets or exams or office hours, whatever, but I saw a lot of misconceptions last semester about P53. So I don't like, I know in my bio 151 class we didn't talk about this, some of them do. P53 is a tumor suppressor. So what that means is when it is functioning correctly, its mechanism is to inhibit at some point in that pathway cell proliferation. So P53 doesn't cause cancer. It's a tumor suppressor. When it's mutated and it doesn't do its function, then we don't have this inhibiting mechanism on proliferation, and that is when we see cancer happening.
Which is why, so that that misperformance or that inability to perform its tumor suppressor role leads to overproliferation because it is uncontrolled of damaged DNA or any of the other examples I already cited.
Is P53 only in tumor?
I think that it is activated and responds during G1 to DNA damage. I could be wrong about that, but you could ask Laura. I think what's most important is knowing what it does and how it works.
Question?
I'm just gonna ask, like, how quick to like find a mutation? You're like, something like this phase or something like that, and you have to go to like 52 days.
So that is what she just asked. I want to say that it's primarily active during G1, but I could be mistaken about that. So that would be a good question for Laura. But again, like if you're comfortable with what it is, what it does, the mechanism, that is what is most important. So, yeah.
All right,
Okay.
that's our slides.
Is that it?
Yeah,
Okay,
that's
yeah,
it.
this one was pretty brief because it's like cell cycle, but yeah, absolutely questions if you have any.
Basically, why is it created if it's inactive?
Why is it degraded?
P53, if it's inactive.
Oh, so I don't think that's P53. I think that's earlier when we were talking about our. Oh,
Once produced is heavily regulated or degraded.
oh, yeah. So that is kind of, that applies to a lot of proteins in the cell. It's a tumor suppressor, right? So if we have active P53, it's going to prevent cell proliferation. So if we don't have any DNA damage, we don't need it around. And I'm sure we also don't want to risk something going wrong and inhibiting proliferation when everything is in order and we need to have the cell divided. So yeah, that's how it works for like a lot of proteins. If we don't need them, we don't want to have them taking up space. Yeah, of course.
All right, I'm gonna stop the recording now. We're gonna stay around for questions.